have some very good values, which I'll discuss in just a moment as well. My style of presentation is please do ask questions during the seminar. This is an educational event. If you have any questions, you don't have to hold us. Just raise your hand. We will be available afterwards for questions as well. I will say jokes during the seminar, so please laugh if you find them funny. If you don't find them funny, just laugh for my confidence. I have given this seminar about 400 times around the world over the past 3 years, so most questions I would have answered, but if you do have a question, go ahead and raise your hand. I will most likely say, great question, I've applied for that, because I've done this so many, many times. As long as you fill out the sign-in form or whatever, I will send you a copy of my slides when I jump on a plane later this evening. So as I mentioned, we are looking at pathway-focused expression analysis and a complete experimental system from sample preparation through data analysis. We have these systems for gene expression, microRNA expression or miRNA, DNA methylation, DNA mutation, cytokine expression. This is ELISA arrays. We also have reporter-based systems. These are signal-based product lines. Plasmid or lenti-based, GFP or luciferase signaling. We also have things that knock down certain aspects of certain processes or pathways with siRNA, and then you can look at the regulation of the gene with some of these products. And also, if you're studying ubiquitin-protein stem cells, we have pathway-focused panels for that as well. What I'll highlight today is gene expression analysis in terms of the workflows and concepts, the chemistry. Everything that I discussed about the gene expression follows through for microRNA expression, DNA methylation, and DNA mutation. There's a couple of modifications in chemistry. Basically, the concepts are all the same. And the concept in terms of sample preparation through data analysis allow you to focus on your pathway. One of the proud things I can say as now a QIAGEN company, a QIAGEN employee, is traditionally you'll put whatever sample, your biological sample, into a pink or purple tube. In a QIAGEN kit, RNA easy kit or DNA easy kit. You put in whatever sample, you get out purified nucleic acid, very high quality nucleic acid. With my system, from SuperArray or SA Bio, you can put in whatever sample and fill into the RNA easy kit, but you get publishable quality data at the end. You can simply take a print screen from our free data analysis software suite, put that into your Word document for a grant application or for a paper submission, and we've seen over 1,200 publications with that. With data resulting from our arrays. Also, I've seen them in numerous grant applications. I sit as one of the technical experts on the NIH study section, and so I can actually review the grants when they do come in to see if they do match the data that you need. So, first off, gene expression regulates biology. So it doesn't matter what area of research you're in, if it's in cancer, cell cycle, neuroscience. Some of the people we've met with today are studying various aspects of biology, but they're all regulated at the gene level, and all of them require molecular signaling for action. So this is a small picture of the EGFR pathway, signaling pathway. Traditionally, if you're studying a signaling pathway or process, you might focus on one gene or two genes. My graduate work at the MD Anderson Cancer Center was on HER2 overexpressing breast cancer cell line. I look at HER2 exclusively as it relates to apoptosis, angiogenesis, cell cycles, cell adhesion, and so forth. But this is 15 years ago. For EGF, I just look at EGF, the EGFR receptor, MAPK, and some others. I look at straight signaling cascade. But when we're studying cancer, we're looking at anomalies in the process. So you want to study all of the things that are related to that process because it is cancer. Something is screwed up. So we want to find out, it might be at the EGF level and something down here, and then something down here. But if we're just focusing straight up on the signaling cascade, we're going to miss out on these other participants in these signaling families. And again, as I mentioned, biological markers define processes. So if you're studying cell cycle, angiogenesis, or inflammation, you might focus on individual participants. And this, again, is my own story. When I was a graduate student, when I looked at new blood vessel growth in my cancer cell line, in my cancer models, I looked at VEGF exclusively. My mentor was a big fan of Dr. Judith Logan, and so we focused on VEGF, because that's what he did. And this is 15 years ago. But in angiogenesis or new blood vessel growth, you'll have VEGF, you'll have influences of VFGF, you have angiopoietins, you have PDGF, you have a variety of other factors. In addition, for cell cycle, you might focus on P53 like I did. But there's other markers in the cell cycle that regulate the cell cycle that are screwed up in cancer. And that's why you have unregulated differentiation and proliferation. And so that's why if you just study P53, you're missing out on MDM2, cyclin, the cyclin-dependent kinases, the inhibitors to the cell cycle. And that's something that you have to study in order to give a full story of what's going on in your sample. And that's why we have stressed at SuperArray the pathway-focused approach. So instead of just looking at VEGF for angiogenesis, you're looking at VEGF receptors, you're looking at FLT3, you're looking at all the downstream signaling modules and things that are to the left and right of it, of the signaling cascade. Because as you do, you'll build a better story. The better story you build, the more strong your grant application is, or your paper is. So instead of publishing in the journal of chunks of science and stuff, you'll actually publish in Cell Science or Nature. That was a joke. No smile whatsoever. There is no journal of chunks or Cell Science and stuff, so that might be a business plan for later on. So again, you want to focus on a pathway-focused approach. And as opposed to a microarray, which is our predecessor product, that gives you whole transcriptome analysis. But that's just way too much information, and you're studying a particular process at a particular point in time when you're doing your analysis. So I did all three areas in my dissertation. But one day I looked at cell cycle, one set of projects. One set of experiments was on angiogenesis. They weren't all looking at the same thing. In addition, microarrays have a dynamic range of about 3-fold, real-time expression analysis of 8-fold. In addition, the data is always focused on a pathway-focused array versus a microarray. So while you isolate your RNA from your sample after you run your experiment, your sample contains, your little tube, after you run an RNA-easy kit, contains all of the RNA that is expressed in that sample. And so that's, there's 20,000 genes. You might have about 32,000 transcripts because each gene can have multiple transcripts. And so how do you assess the expression of different messenger RNA in a sample involved in a specific process like apoptosis, angiogenesis, or cell cycle, and compare it across multiple samples? Because again, this is always differential expression analysis. You're looking at breast cancer versus non-breast cancer. You're looking at treated versus non-treated. You're looking at time courses. You're not looking at absolute values. Yes, sir, no, it's not. You're actually looking at the differences between, again, sample 1 or condition 1 to sample 2 or condition 2. And of course, we have an answer for that in the RT-squared profile or PCR array. It's a very simple and accurate pathway expression analysis tool. And our product has been on the market for about 7 years, and our product has been used by a variety of researchers spanning all realms of life sciences research. And in the past few years, we've actually expanded to who we sell to in terms of the molecular biology applications that researchers are doing. I was in Montreal, and I spoke at the Canadian Beef Council. And those folks are actually using our product to look at developing cows that have better marbling patterns in the beef. Better marbling means tastier meat. And they're doing this at the gene expression level. So there's some other researchers looking at malaria research that we met yesterday here at UC Davis. There are researchers doing wheat, cotton, rice. And again, they might do it at the protein level, but ultimately it's the gene expression level that they can look at their differential expression analysis. Now, as I mentioned, we support all biomedical research. We have over 150 pathway-powered PCRs available, spanning a variety of research areas. We have areas of research such as cancer, such as diseases, processes, such as apoptosis, and categories of pathways such as inflammation. And this, as I showed you earlier, this is a basic 96-12 plate PCR array. It has 96 wells. We have 84 genes per pathway, plus 12 controls, which I'll discuss in a moment. And so the reason we have multiple pathways in each category of pathway is simply, I'll just give the most simple example. I take the table. I will have an inflammatory response in my left leg. I will not have that response in my I will have an inflammatory response in my right leg and not in my left leg. So I want to look at the differential expression of this inflammatory gene in this right leg, or this right foot. And so when I kick the table, I'll get an NF-kappa B signaling pathway response. I'll get an inflammatory cytokine receptor response. The one thing I won't get is a toll-like receptor response, because that's more of an infection sort of deal. And so in infection or inflammation, there's so many genes that we simply put specific genes, specific pathways on the array. And we did this so that you can identify specific pathways that you might want to study first. So now that I've done my analysis, done my experiment, I'm taking the table and all, and then I see that this gene, and this gene on this specific pathway were differentially modulated. So like these three were upregulated in this light versus in this light. And so then we want to validate those results with more biological references as a sample. And so I want you, and you to all repeat the same exact experiment and see if we get the same biological result. And for that, we actually have custom PCR arrays. So you can take those 3 genes, or 5 or 10 genes, or whatever, or your own favorite gene list, and make a custom array. And we have it available on the 96-well platform, the 384-well platform, the 100-well disc, and the Fluidine Biomark 96x96 disc. And we have wet bench validated assays for every gene in the human, mouse, rat, cephalopoid, recent dog, and pig genome. And if you're doing some other genome, we do require full genome annotation as well as full transcriptome annotation by the NCBI. And we just recently released pig just a couple weeks ago, and we're on the way of going to cow and dog. But that's just because NCBI has uploaded their databases, and so we do need that information. We do have custom capabilities where we can expand out to something else, but just let me know. One thing I always like to highlight is we have 150 pathways, but if your specific pathway idea isn't on our list, let me know. If you're the world expert in foot-kicking table disease and you don't see that on our table or our list of arrays, let me know. And I can say this honestly because this is actually this epithelial mesenchymal transition. That is my pathway. That is pathway number 90. That is a result of my graduate work because that was my dissertation. I was at the company for about a year. I knew I was going to get fired after about a year. Joke. And so, man, you guys, come on. And so then I said, hey, where's my pathway? If I was a researcher, I would use this one. And so this is now one of the top 10 selling arrays. In addition, the molecular toxicology pathway finder array. This actually resulted from a collaboration that we had with Novartis, Pfizer, Merck, and Lilly. As they were screening their drug compounds, they came to us and said, hey, we keep buying these custom arrays. Can we work with a consortium? Can we come up with a gene list? So they worked with our scientists, our content development scientists, and we made this pathway array. And so not only are the pharma companies using it, a variety of researchers in academia are using it as well. And if you have a pathway idea, again, let me know. I have this wonderful thing that keeps buzzing and blinking. It's a global BlackBerry, so wherever I am in the world, I have access to you guys. So if you have a question, you have a comment, you have a pathway idea, let me know. I'll tell my content development scientists, and then we can hopefully help you out with that. So let's look at an actual array. So this is a basic 96-well plate array. This has 84 pathway-focused genes of interest. This is the human inflammatory cytokines and receptors PCR array. It has 84 pathway-focused genes of interest, plus a control. There is nothing magical about this product, except the value that we provide in terms of the control and the system. They're just validated primer assays. So they're not just bioinformatically validated. We actually wet bench validate. So we actually test every assay and every lot of every assay to guarantee that it works when it leaves our building in Frederick, Maryland. We guarantee it works. So if you are designing your assay, I beg you to stop. We've already done it. You're just reinventing the wheel. We have done it. We have wet bench validated, and we provide you that wet bench validation data in the specification sheet that gets mailed with every assay or with every array. And I'll go over this layout in just a moment as well. But as I mentioned, every assay it undergoes a melting curve analysis. This guarantees that each assay produces a single peak on a melting curve. This shows that each product, PCR product after 35 cycles, is of one melting event. You will not see primer dimers, which usually appear at a lower melting temperature. You will also not see double peaks, which might indicate another melting event, which would mean that there's some non-specific binding, and that primer can then make something else. This is one melting event each and every time. In addition, we provide PCR efficiency, and all of our PCR primer assays are guaranteed to give you about 95 to 99% PCR efficiency. And why is this important? For staining 96 genes potentially, or 384 genes at a single time. Or in fact, you could be looking at a 20,000 gene array in one run. And you want to make sure that all assays work at the exact same rate. Because if they don't, since they're doing differential expression analysis, you'll get funny numbers that don't mean anything. And I'll highlight this particular aspect in just a moment. To highlight the fact that we have every assay for every gene in those 7 genomes that I mentioned earlier, this is a screenshot from our website at sabiosciences.com. Simply type in whatever gene, and we'll have that assay to you in about 3 days. They are synthesized and manufactured in our facility in Frederick, Maryland. We built the building right next to a UPS distribution site, so they pick it up at 6 o'clock every day on the dot, drive it across town, get on a plane, and they're in your